Results. The results of all of the articles we selected were similar, showing weak to no association between caffeine consumption and the risks of dementia. Additionally, after collecting and analyzing the data, the researchers all concluded the same significant issue, that there was a substantial number of confounding factors involved. The 4 articles explained the potential benefits coffee has due to its antioxidant agents. However, they failed to discover if caffeine was a neuroprotective agent. With such similar results, it is unsurprising that information for all 4 articles was obtained from the same types of sources: longitudinal studies such as cohorts, cross-sectional, and case control studies. Only Grasso et al. Also provided a small amount of evidence from randomized control trials, which Kim et al. Also concluded would be beneficial in future research on the subject. While each article measured something a little different, the articles were all comparable in the general goal of their research, the time frame being studied, and in their results. All 4 articles were based on the interest in the possible preventative effects of caffeine, specifically caffeine from coffee, on cognitive impairment in individuals with no current signs of cognitive decline. The researchers all used similar search terms to collect and provide data from observational studies over a similar extended period of time. They all presented their data in a similar fashion, using the heterogeneity model with confidence intervals, case control studies, cohort studies, and cross-sectional studies. They used graphs to present the data, which provided nice visuals showing there was little correlation among caffeine and risk for cognitive decline. The common confounding factor between all of the studies that proved difficult for everyone to work around was that chronic smokers tend to drink several cups of coffee in a day, adding tobacco and nicotine to the increase in daily caffeine. Overall, our results on caffeine consumption and risk for cognitive impairment, as well as those of each article, were inconclusive. The human brain is extremely complex, and genetic precursors to Alzheimer's and dementia are still being discovered or are unknown. And with several modifiable risk factors influencing individuals at different regularities, measuring the long-term effects of caffeine consumption becomes a daunting research endeavor. Ultimately, the researchers discovered there needs to be more epidemiological evidence in order to understand this phenomenon. For example, since coffee and tea were being used as the source of caffeine, rather than caffeine on its own, there was no way to separate the results of the caffeine versus the considerable antioxidant agents in the beverages that have previously been linked to decreasing the risks for dementia. Additionally, since most of the studies used for all 4 of the articles were observational studies, the amount of caffeine and confounding factors were unable to be controlled.